Assalamu alaikum everyone. Today the topic of my video is what is research, how to conduct it, and what are different approaches and methods that can be used to conduct a research, and what should be the characteristic or the traits of a person should be if he or she is conducting research. Research is basically part of your life, also known as holy curiosity, which Einstein termed that every person is curious about everything around you. So, when you are conducting research on academic level, you must be aware of the fact that you need to critically analyze everything and you must find something new from already existing data that is available in previous literature. Another thing you can do is try to find something that was missing in one of the previous research work done, and you can work on it in order to improve your results or obtain a better outcome. So let's, for example, you need to find a cure of some disease. Let's take example of cancer. Now you have to focus that first of all you need to review the literature. I mean, if you are conducting research either on MPhil level or PhD level, it doesn't matter because the more important thing is goal. Degree is not the obstacle over here. You need to focus on your goal. If you are, it can be anything else. I mean, it can be related to agricultural field. It can be related to fertilizer, biofertilizer, biopesticide. It can be anything from biocorrosion to bioremediation. It could be anything. If you are focused on that goal, you need to first of all you need to be clear on that topic, that you want to do research on that topic. Then first thing is review the literature. You can do literature review from credible websites, and every topic can be found on these websites, from ranging from pathology to veterinary, zoology, everything. And these websites are mostly NCBI, PubMed, and all that, Springer, SearchGate, etc. You can easily find, but you need to focus on research articles. You need to study their methods. You need to learn. You need to understand how they have conducted research, what methods did they use, and how did they statistically analyze those results they obtained. And then you need to analyze the differences, if there is a difference in the protocol they used. I mean, it is possible that in one article the focus on method was different, and the second article they used some different kind of protocol. So you need to analyze that protocol as well, so that you can decide later on that what kind of method you need to obtain a better result in your research. For example, if I want to know the quantity of flavonoids in a certain plant extract, I can do it in two different ways. Either I can use sodium hydroxide and let flavonoids react with sodium hydroxide, and yellow color will develop. And then I can use a colorimeter, the visual spectrometer, and then read the absorbance. And second, I can also do it without using sodium hydroxide and just do it on UV spectrometer. So basically both of these protocols will tell me the same thing, quantity of the compound I want to find out, but there should be a certain standard which can help me decide which protocol I must choose. In this example, I will go for sodium hydroxide protocol. The reason is that it will be more specific as compared to UV, because HPLC was not conducted, and plant extracts, the flavonoids in that extract were not separated. So there is a possibility that absorbance value of certain wavelength might not be only indicating the amount of that flavonoid. It can be indicating some other compound as well. So that is why I will select the other method of sodium hydroxide. So this was just an example. Another example which I can give is obtaining number of bacteria You can either go for optical density or you can go for plate count method. I mean, you must decide which one is more accurate, more efficient, which one is easier for you. I mean, most important thing before starting a research work is, I mean, you will be doing it, that first of all you need to analyze, do you know that protocol, can you really do it? You need to also understand that, is that protocol cost effective? I mean, you must focus on saving finances and obtaining better results. You really not need to waste your money. And one more thing, before starting research, you write synopsis, and before writing it, you need to understand one thing, that when you have written some kind of protocol, if you have mentioned some kind of protocol in your synopsis, you need to do it afterwards, no matter what. So you need to understand that if you have written, for example, NMR, HPLC, GSCMS, some kind of procedure related to DNA sequencing and all that, you need to understand that, can you do these procedures in your own lab, or do you want to do it in some other lab? I mean, there are many things which are not available in our lab, so you will have to find it somewhere else. In order to do that, you need to find someone who can help you, uh, help you do research in that lab. So you need to do planning before writing synopsis. You need to know where NMR is, so that you can do it afterwards easily. You will not face problem at the time of thesis. You will not be running here and there and asking what to do and how. You need to plan it beforehand. Now let me tell you some good qualities of a researcher, that how a researcher should be. The first and most important quality that a researcher should have is a neutral approach. Now, what is a neutral approach? For example, you find similarity between two bones, and you decide, give a conclusion that similarity between these two bones mean that it is a common ancestry. It is also possible it is just a common design. So the point I'm trying to saying is, I'm trying to convey is that you must be a neutral. You cannot be biased. If you are biased, then it is not science. Science cannot be biased. You need to properly analyze each and everything, then give a conclusion. Secondly, you need to be very good in observation. If your observation is not good, then it is possible that you might miss everything. If you are a very good observer, you will observe very small little changes or very small pattern, and you need to note down each and everything going on. And the third thing is that your problem statement must be very precise and very, you must obtain good amount of data for that problem. For example, if you want to treat a problem of protein deficiency, then your introduction literature view must be accordingly. Your introduction must indicate clearly that what is your problem and how you are doing research and how will you do it, and literature view will be accordingly. Fourth thing is that you must allow your mind to explore things. It is, the main most important thing of a scientist is exploration. If you can't explore, then how is it possible that you are a scientist? I mean, it is not possible. You really need to be creative, and exploration is the most important thing. Without exploration, I mean, if you are just worried about contamination, remember that contamination is the reason that that we are using antibiotics today. If there was no contamination, I mean, how is it possible that we are using then antibiotics today? So don't be negative. That's the fifth most important thing. You cannot be negative. I mean, if you are not obtaining anything, that is also a result. You must stay positive and try to understand that it is not possible that what you think it should be, it is not possible that it will always be that way. Result can be different from your expectations, and you cannot force your results to be obtained in a certain way. So that's it for today. Stay tuned, like, share and subscribe for more videos related to such topics.